organism would be the result, okay? I don't know what kind of organism would end up being the 92 chromosomes, all right? Now, it might be interesting to see, but the cell would basically just quit, and the organism would die, all right? So, at the end of my S phase, all of my chromosomes have duplicated. In this cell, I've got other organelles, all right? I've got, endoplasmic reticulum. I've got Golgi apparatus. I've got vesicles. I've got ribosomes. I've got all these other organelles. Well, because the cell has basically decided at this point right here, hey, I got to split, okay? It wants to make a copy of everything else. So it's going to make a copy of everything else in the cell. It's going to make copies of proteins that are in the cell membrane. So by the time we get to the end of interphase, get to the end of G2, this cell is very packed, okay? So now it's like, okay, this is it. I'm out of here. And mitosis begins. And we start prophase, metaphase, anaphase, telophase. If I ask you, what are the phases of mitosis? This is what you tell me. You tell me prophase, metaphase, anaphase, telophase. Your textbook, for example, is going to talk about something called prometaphase and stuff. That's a waste of my time. Okay? Prophase, metaphase, anaphase, telophase. Something happens to the DNA at every stage. This replication of the DNA, where I have taken every single strand, made a copy, and have now 92 or 46 pairs, they are now being held together in what we think of as the classical X for chromosomes, where one half is the old strand and one half is the new. The job at this point, because we have now created this entire copy, the entire thing, some of them millions of base pairs long, and we now have old, new, and we are now looking at what is called chromatids, and they're held together by a centromere. Does everybody remember that? So, at this point, we are going to find that for every stage of mitosis, something happens to these chromosomes. One of the things that will happen in that cell, that centromere, that little tube-looking structure, okay, duplicated itself, and now it's going to begin to pull apart. One to one end of the cell, one to the other. The stages of mitosis: prophase, metaphase, anaphase, telophase. In prophase, with prophase, in the nucleus of the cell, the DNA begins to, it's duplicated, and it begins to take on the X shape. Also, in prophase, that nucleus is beginning to break down. Okay? So, prophase, When you guys were looking at this under the scopes, was it noticeable? Was interphase, where it looked like the stuff was just spaghetti inside the nucleus, where it didn't look like it had any shape or any of those spindle fibers or anything like that, was that very identifiable under the scopes? Okay, so when you were looking at it under the scopes, it should have looked just like the pictures in the book, where interphase just simply looks like you've got a very well-defined nucleus. You don't have any strings or anything looking like it's showing up, okay? But as we begin to enter prophase, you begin to see those spindle fibers, and they're pulling to each side in metaphase. Metaphase, middle. Okay? Because those X's are now attached to the spindle fibers, and they are completely lined up in the middle. What is the purpose of that? What is the purpose of having that X where I've got an old strand and a new strand, and it's held together by a centromere, and it is now attached to a spindle fiber?
I don't remember what it's called. They come apart equally.
They're going to come apart.
I don't remember what it's called, but like, so the new one's here and the old one's here, and they, like, the spindle takes this half of the X and the other one takes this half of the X, so they get the new and the old.
So, semi-conservative. That's what it's called, because we have old and we have new. Now, what's going to happen? Because this is metaphase. They are attached to a spindle fiber. Those spindle fibers in anaphase. In anaphase, we don't have any more of the nucleus present. The spindle fibers are beginning to shorten. As they begin to shorten, they are going to pull chromosomes apart. So we end up like this. With half of that X going to each side. Now, once we have anaphase, the next part that comes along is telophase. Telophase is going to be where I now have two new cells. Telophase 2, T and T, okay? They begin to form another nucleus. Those DNA begin to unwind, all right? And we now have the two new cells, and then the next thing that comes into play is going to be cytokinesis. That is not a process. That is not a phase of mitosis. The definition of that is the division of the cytoplasm. The job, take that cell that was one, now has copies of everything, and begin to pinch it in, pinch it in, pinch it in, until I have two cells. This is going to be very important for the topics as we go through them. The next chapter that we're going to start, it's a little weird, okay, because we actually cover chapter 26 before we move to chapter 5, which is the integumentary system. But the reason for that, chapter 26 is about the digestive system, all right? And everything that we've talked about the utilization of the fats, of the sugars, of the proteins. We've done that. We're going to connect that information to what takes place in the digestive system. So we'll do 26 next, okay? And this cell division, remember, there's only going to be two areas where it doesn't take place. The nerve and the muscle, okay? So that is going to be a topic that we have to remember as we move forward. Because if you think about it, cell division is so important that if something goes wrong with it, it can result in cancer. Because cancer is simply out of control cell division. Which is a tad bit interesting. But bye. I'll see y'all on Tuesday.